Hola, welcome to Chapter 6, Microbial Nutrition and Growth, Part 4. This is the continuation of microbe media, or things that microbes like to grow in. This video talks just about special culturing techniques. So, for example, my favorite microbe, because the name is so cool, Treponema pallidum, that causes syphilis, can't be grown on an agar plate. It can only be grown in these innocent little rabbits. That's terrible. Mycobacterium leprae, that causes leprosy, can only be grown in this creature. So, if we want to study microbes, we have to grow them. We usually can just, well, not usually. Sometimes we can just grow them on a plate in the lab. Sometimes we have to use animals. So Mycobacterium, for example, is grown in an armadillo. Viruses, small bacteria like rickettsias that cause Rocky Mountain spotted fever, and chlamydia, which is the most common STD, but I think they're called STIs now. I think they took the D out, so they're sexually transmitted infections. They need to grow inside cells. So you could grow them inside a bird egg, because that's a cell, single cell, or you could use a culture of living cells to grow them. And here's some eggs being inoculated with viruses to grow them. If you want to grow certain microbes that don't like a lot of molecular oxygen, so low molecular oxygen, not no oxygen, but low molecular oxygen, for organisms that require low levels of molecular oxygen, you can make these kind of, I don't know, they seem like you could just make these pretty easy. It's an incubator. It's got high CO2 and low molecular oxygen. So it looks like it's just a big old glass container with a lid, and then you burn a candle inside it, and then here's your plates that you can grow. So organisms that like a lot of CO2, like Neisseria gonorrhea, you can grow them in these special culture containers called candle jars. Enrichment cultures that you can find microbes out in the world that are present in really, really, really low numbers, or those microbes that are just outgrown by other bacteria all around them, so it's hard to isolate them. So you use these things called enrichment cultures. So you use selective media, so you just are selecting for that desired microbe in that population of microbes. How do you preserve cultures? You could use refrigeration. That slows down microbial metabolism. That way you can preserve microbes. So let's say you're studying them in a lab and you want to preserve them. You could use deep freezing. It also decreases the microbial metabolism. You can, with deep freezing as opposed to refrigeration, deep freezing is for long-term preservation of cultures. And then there's lyophilization. So that's freezing. It's actually drying the microbe and then freezing it. It also decreases the microbial metabolism. And you can store your microbe of interest for decades with lyophilization. And here's a lyophilized sample in a vial that's been dried and frozen. If you want to study an organism, you have to isolate it, and then you have to be able to culture it or grow it. So you take an inoculum of your sample of interest with the microbe in it, and then you inoculate it into this medium. So that's all where all the nutrients are that the microbe would like, and then it can grow into a culture. So you could have a broth culture, that's liquid medium. You could have an agar culture, that's solid medium. We covered that in previous chapter 6 videos. You can grow colonies on that agar, and then your colonies growing on the agar. So remember, colonies are, a single colony could have 1 million microbes of interest in it. Colonies have really distinctive colors, they have shapes, they have sizes, elevations, textures, and very distinctive margins. So for example, this is, so here's the plate. This is a colony of Serratia marcescens, and it's definitely red, so the color is red. It's circular. It's convex as opposed to concave. It's large. It's smooth and shiny and opaque. So if you go on to be a microbiologist, you can get really good at describing cultures and the colonies that grow on the cultures of bacteria. So here's a diagram that shows the different kinds of shapes you can have in a colony. So you could have a circular shape, that's pretty easy, and then all these other shapes. The margins could look like this, so that the margin of the colony could be an entire, or it could be an undulate margin. The elevation, how high up do they go? Are they flat, or are they convex? Do they just have a little raise to them? You could be a punctiform colony or a large colony. Your texture could be smooth or rough. The appearance could be shiny or dull, could be pigmented or non-pigmented, and optical property, it could be opaque, means you can't see through it. It could be translucent or transparent. And again, here's a photograph of a light micrograph of Serratia marcescens, a colony of Serratia marcescens. So again, it's really important to note, this is not a single cell of Serratia marcescens. It's a colony of Serratia marcescens. You could have, hundreds of thousands of single Serratia marcescens bacteria in that colony. Clinical sampling. This is a table from your book, Clinical Specimens and the Methods Used to Collect Them. You probably already know this one. The type of specimen, blood, you use a needle aspiration from a vein. You take blood from a vein. In the transfer tube that you put the blood in, you have to have anticoagulants in there so the blood doesn't coagulate. If you want to get a specimen from the stomach, you use the collection method of intubation. If you want to get a specimen from the lungs, you could get the sputum that was dislodged by coughing, or you could use a catheter. The diagnosis and treatment of a disease depend on the ability to isolate and the correct identification of the pathogen of interest. So you, as a clinician, you need to prevent contamination of the sample, and you need to prevent infecting your cells with the sample from the patient. And the samples can be anything from feces to cerebrospinal fluid to blood. And you have to transport the samples quickly to the lab so the pathogens don't die. It's trying to figure out what the pathogen is. So this slide talks about obtaining pure cultures from these things called colony forming units. So a colony forming unit, it's just a cell or two cells or several cells that form a colony. So if you, in this part of the cartoon, you have one cell, you allow it to grow, it forms a colony. Here you've got a couple cells that are probably the same type of cell, they grow and form a colony. Here's several cells, probably all the same type, they grow and form a colony. So these guys would be the colony forming units on the left, and then the colonies would be on the right. So the clinical specimens contain the patient's pathogen of interest that you're trying to figure out what made the patient sick, and it also includes the patient's normal or resident flora, aka the patient's normal or resident microbiota. So when you get the specimen from the patient, you want to isolate a pure culture of the suspected pathogen. So that, in other words, you want a culture composed only of the suspected pathogenic cells, that arose from the one pathogenic cell or a group of related pathogenic cells. So in this part of the cartoon, this would be a group of related pathogenic cells. This pathogenic progenitor cell is called a colony-forming unit, or a CFU, and it will produce one colony. So from these, let's say, 4 pathogenic progenitor cells, you form one colony on the plate. So how do you obtain these pure cultures? You do a streak plate. So here's your hand. Here's the inoculating loop. So you went in, you dipped, so you've got your test tube here with the suspected pathogen in it. And you take your inoculating loop after you've sterilized it in a flame, and then you let it cool. Then you dip your inoculating loop in, you collect a specimen, you collect the sample of the specimen of interest, and you start here, and then it shows how you streak these plates. So you take your inoculating loop and you go back and forth and back and forth and back and forth and back and forth. Now, if you just plated this part, this number 1, you wouldn't be able to see the individual colonies. So then what you do is you go back and you incinerate your loop again, but you don't dip back into the culture. You just pull down a few microbes, or a few pathogenic microbes from here, from the first streak, and then you go back and forth on the second streak. And then you re-incinerate your loop and let it cool. And then you come back and you pull down some microbes from here. And then that's 3, and then the 4th that you do, you should have your microbes strung out enough, see how you're streaking that you can get these individual colonies. So here's an individual colony. Like I said, if you only stopped at this first pass, you can't see any individual colonies here. You can start to see individual colonies here, but in this 4th pass, you can really see the individual colonies. So then you can decide what pathogenic microbe you have. Okay, this is called the streak plate technique to isolate individual CFUs, or colony forming units. And as I said, the inoculum, that's when you put your inoculating loop and get some sample, that's an inoculum. You gradually dilute it to a point where you can see colonies from a single colony forming unit. And then you can examine the colony morphology. This is a good technique for culturing aerobic organisms, because obviously they're going to be in oxygen, so they have to be anaerobes. You could also use this other technique where you obtain pure cultures. So what you do is you set up your tubes. So you set up the 1/10, 1 to 100, 1 to 1,000, 1 to 10,000, and you put 9 mils of media in each one of these tubes. And then you go get your original stock, which is called 1, and you take 1 mil from your original stock. So your microbes are in here in a media. They're undiluted. You take 1 mil out of the undiluted stock, or the full strength stock, and you put it in 9 mils of media. That is a 1 to 10 dilution. Then you take 1 mil out of your 1 to 10 dilution and you put it in the next 9 mils of media. That's a 1 to 100 dilution. Then you take 1 mil out of the 1 to 100 dilution and you put it in 9 mils of media. You get a 1 to 1,000. Do the same thing, you get a 1 to 10,000. Oh, and I should note, so you take your first mil out of the stock and you put it into this first tube. Before you take 1 mil, actually before you take 1 mil out of the stock, you want to vortex it. So you shake it up so all the microbes haven't just settled to the bottom. So you want to vortex it, or it's a type of mixing, so that you can get the microbes so they'll be throughout the stock. So when you take a mil out of there, you're getting a good sample. So the microbes are separated using a series of dilutions that I just talked about. And then the diluted inoculum. So these guys here, the 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000. That's the diluted inoculum. It's mixed with cooled agar, and you pour it in a plate, and then you can look at the colonies that are living on the plate. I think that's it.